Good afternoon, everyone. So if you can hear me, can somebody just unmute and tell me yes?
Yes, ma'am.
Good afternoon, ma'am.
Okay, Lakunle, thank you. Thank you.
Yes,
All
ma'am.
right, so you are welcome. If you graduated from Unilag, I welcome you back. Nice to have you back. If you did not graduate from Unilag, I welcome you to the University of Lagos, the University of First Choice, and the nation's pride. So welcome. This is
Thank you, ma'am.
MIC 813. I'm not sure how many of you intend to offer this course. I'm seeing just 7 participants. And on our platform, I have 8. Usually, the least number of students registered for that usually take this course is between 20 and 25. But I understand that some people have not resumed. Olakunle, is that true?
Yes, ma'am.
So if we start lectures, have lectures started? Have other lecturers started?
Yes, ma'am. We've had 2 different.
You are cracking. I cannot hear you. Oh my God. So at least I can somebody. Does somebody else want to speak? Olakunle is cracking.
Good evening, ma'am.
Yes. Good afternoon. Adetono. Abi, who is speaking now?
Goodness, ma'am.
Goodness, how are you, Kukoyi?
Fine, ma'am.
You guys have had one other lectures, right?
Two.
2. Because my
Eight,
worry is, if we start lectures now, then other people resume. What's going to happen? They're on their own, they will read, right?
I think so. I think they will catch up, ma'am.
We will have to catch up. That's true. All right, so we can start then. Olakunle, you have to stop talking. You are still talking. Oh. All right, so welcome. Let me have a look at my participants here. My own name is Dr. Olanike Maria Buraimo, and I am in the environmental microbiology unit aspects. My interest is on bioremediation first, then I'm into bioconversion of waste to value-added products. I also venture into antimicrobial resistance and air quality management. So that is all I do for now. So I can see Adebola Ogunleke, Chukwuneme Raphael, Ekwe Muka Daniel, Goodness Kukoyi, and Osofi's son, Boluwatife, and of course Olakunle, I guess. So I welcome you all. And this is what we are going to be treating. You can see my slide. We are going to cover microbiology and biochemistry of agronomically important soil processes. Central role microorganisms play in soil biogenesis and protection. The importance of biological equilibrium and significance of soil microbes to environmental quality. Evaluation of the biotechnological and commercial potential of microorganisms in sustainable agriculture. We are going to look at plant microbe soil interaction and functioning, commercialization of soil microbes, and environmental aspects of biocontrol. We'll look at some case studies highlighting classical and GM crop development and application of soil microbes like we have the biofertilizers, the mycorrhizal fungi, as to how they are related to sustainable crop production. Then we'll look at molecular genomic techniques for identification of soil microbial community structure and dynamics. So for these courses, we are going to do it together. This is an MSc class. So it is not for you to sit down and be spoon-fed. I will take 2 or 3 of these topics, and then you will do yours as term papers. After putting your work together as term papers, then you will come up to do your presentation. Previously, it used to be 3 people per topic because there were many. But as it is, you may likely have 1 person per topic, so brace up. I'm going to think about, usually with ballot, I give topic without bias. I will try to see if my PhD student is back from his holiday, so I can send topic to him. So you can go to him and pick your topic by ballot. Then you send your topic to the WhatsApp group with your name, your matric number, or Olakunle will coordinate, put everything together in a table. I will let you know. So you have to go to school to ballot. You let me know when it is convenient for you between now and Friday if you are going to be in school. Let me know when you will be in school. So you go and ballot, pick your topic, and I'll give you 2 weeks to prepare your topic. And you will also ballot on when to present. On presentation ballots, if you pick 1, that means you are the first person that will do the presentation. If you pick 5, that means you are number 5. And we are going to be having 2 presentations per week, so that in the next 6 weeks, you are done. If I'm able to give my own lectures, I've prepared lectures for 2 weeks already. If I'm able to prepare another 2 weeks, then the remaining 6 topics or 7 topics, you pick it one by one. Then you will do your write-up, term paper, and presentation. So that will serve as your CA. I'm supposed to teach this course with Professor Ilori, but as you all know that he's been very busy for the school, it's part of the university management. He was formerly the director of maritime. So since we've been teaching this course together since 2020 or 2021, I've been the only one teaching on behalf of both of us. So I'm braced up for the assignment. Currently is the director of academic planning, so he doesn't even have the time. So I'm ready to take it all alone. So your term paper, your presentation is going to be 20 marks. Then the term paper you will submit after all the presentation, and we do your correction, you will correct your write-up, and then you submit your slide and your write-up. That will be 10 marks. At this junction, please, if you have any question, feel free to unmute and let me hear you out. I'm going to put off my video so that I will have a good internet coverage as we start the lecture. Please feel free to speak. Any question, if you don't ask your question now, I will assume that you are okay with everything I've said. Adebola Ogunleke, are you okay?
Yes, ma.
Did you understand everything I've said?
Yes, ma, I understand, ma.
Raphael. Chukuneme, Raphael, are you fine? So Raphael just put on he joined the class, but he's not in the class.
Ma, I am in the class. I am fine, ma. I am in the class, ma.
So why did you not speak?
I was trying to unmute.
All
Sorry
right.
about that.
Adebola Ogunleke, you said you are fine, right?
Yes, ma.
Okay. Daniel, are you all right?
Yes, ma'am. Fine, ma'am.
Goodness.
Yes, ma'am. Fine, ma'am.
Oluwatife.
Yes, ma'am. Fine, ma'am.
All right, so you all understand. Definitely after you ballot. So since what's his name now? Olakunle has been going in and out. Obviously, his internet is bad. So Adebola Ogunleke, you listen carefully so that you discuss with him, or probably you take the role up. At the end of this class in the evening, discuss among yourselves. Let me know when you go to Unilag to meet my PhD student to ballot, both for your topic and for your presentation time.
Okay, ma.
All right, so I will give you 2 weeks. Once you ballot, I will give you 2 weeks to do your write-up. In those 2 weeks, I will not disturb you at all. You will go to the library, do your literature review, and then begin to write. You know the meaning of plagiarism, right? So
Yes,
don't
ma.
Yes, ma'am.
do plagiarism, because I can turn it in your write-up. AI, I know the AI languages. While I cannot prevent you from using AI, I will encourage you to read whatever you have and write it in your own words. Some of the things that will be used to judge or to score your presentation, I'm not going to do it alone. I will have 2 or 3 PhD students with me so that I won't be biased. Then your mark will be pulled together and I'll find the average. You must have an introduction, you must have an outline. Because you are going to be doing your presentation virtually, you must be audible, clear. There is no excuses that your internet is not good. This, you don't have data. There's no, we're having virtual classes this semester in Unilag. And even if we are not, we can decide to use any method that we like. So you get those things, make sure you are audible, make sure you are well dressed, because we'll look at your face first before you put off your video. And again, I would give you for every topic I teach you, maybe the 2 or 3 topic I will teach you, I will give you your slide to read. Then, once we finish correcting you after your presentation and you make all your presentations correction, you will share your slide on the WhatsApp platform so that your colleagues can have access to it to read. Because from everything, both my own and your own, that is where I will pick the exam questions from. So you don't think I am biased. You have to do a thorough job, because I have all the notes and slide of every topic I'm going to give to you. I have them already. So I know what is supposed to be contained in your presentation. So you can't just come and babooze me. I already have all the slides for this topic. So I know what you are supposed to be presenting. So anything short of that, I know what to score. You must have diagrams. Try to play around with diagrams, with figures, to boost your work, because figures also carries mark. Your references, how current they are, how well you cited them, also will carry some mark. So I hope I've been able to give you some info so we can start the lecture gradually. So I still ask again, Olakunle, you are just coming in, so your colleagues will tell you everything I've discussed with them. So once again, I welcome you. Any question? The absence of no question, then we can start with the first topic. Our first topic is microbiology and biochemistry of agronomically important soil processes. And the outline here is it. We'll start with a brief introduction of the topic. We'll talk about soil microorganisms of agronomical importance, the processes that are powered or carried out by microbes of agronomical importance. Then we'll look at some processes like decomposition, briefly about nitrogen fixation. Of course, you are going to read phosphorus solubilization and potassium solubilization or production of ceratophores yourself. So, what is soil microbiology? What is soil microbiology? Soil microbiology is the study of microorganisms in the soil and the functions they carry out. So people that read soil microbiology study the microorganisms that are in the soil, and then they try to attach the functions. What function do those microorganisms carry out within the soil? What is soil biochemistry? Study of the chemical processes that are mediated or carried out by the soil microorganisms. Because we are talking about microbiology, you know it is not only microorganisms that aid in biochemistry of the soil. We have worms, we have ants, we have some other eukaryotes. But we are microbiologists, so we are dealing with microorganisms especially. And indeed, it has been found that it is microorganisms that carry out majority of the biochemical processes in the soil. So when we talk about agronomic importance, we are talking about healthy soil that supports crop production. So talking about agronomically important microorganisms, microorganisms that support healthy soil, crop production, nutrient cycling, of course, organic matter decomposition, and those microorganisms that enhances soil fertility. What is agronomy itself? Agronomy, people that study agronomy or the agronomists are people that study that branch of agriculture that deals with field crop production and soil management. Most of them deal with what we call staple foods like grains, rice, corn, and all that. So not only with the crop production, but they also deal with how to manage the soil for proper health, fertility, and preservation. So soil, what is soil itself? There are many definitions, but essentially, soil is the surface material that composed of mineral particles, organic matter, water, air, and living organisms that covers most of the surface of the land. So if you take some quantity of soil and you analyze it, you will find different mineral particles, different organic matters, whether it is decaying leaf, decaying earthworm, which is living organism and all that. It's mostly consists of water and air, you know. So for example, sandy soil is more hairy because it is more porous than clay soil. So air and then living organisms like worms, crabs, microorganisms, insects, ants, and all that. So the soil is also the medium that supports plant growth and plays a vital role in various economic processes. Soil differs. They vary greatly in their chemical and physical properties. Like I just mentioned, you have different types of soil. This is not geology class. This is not agri, but we're just trying to mention some of these for you to understand. You can have loamy soil, silt soil. I mentioned clay soil. They differ in their characteristics and properties. And some of the processes that can affect the characteristics of physical properties of soil includes leaching, weathering, and various microbial activity. So all this activity affects the soil that make them to become different soil types. So what are the components of soil that I just mentioned? So we mentioned mineral particles that consist of about 45% of the soil. You have the sand, silts, and clay particles that influence soil texture and structure. So the soil texture will determine the nature of the soil. If I see clay soil, I can say, oh, this is clay soil because it has less hair or space. It is sticky, and all that. It holds water. It does not allow water to filter through. So if you all studied Greek in your secondary school, so you still remember this. We also have the organic matter that is just about 5%. So if you look at the diagram that I showed here, the organic matter is 5% of the whole of the soil. But for the organic matter, you still have different things. You have the humus, which are dead organic matters like fallen leaves that are already decaying, maybe a dead rat, human being, worms, microorganisms themselves that are dying and decayed to form humus, which is the major component of the organic matter, is 80%. We have the roots of plants and all that, 10%, then organisms themselves, 10% of the organic matter. The organic matter is 5%. So if the organism is 10% here, that means the organism is just about less than 1% of the whole soil component. So we have air, 25%, water, 25%, mineral particles, 45%. And these are the constituents. Organic matter have decomposed plant and animal residues that made the humus, then the improved fertility and water holding capacity of the soil. Water is 25%, held in soil pores, essential for plant growth and microbial activity. So if you have sandy soil, for example, it may not have good support for plant growth because it has a lot of pore space, so it does not hold water. Hair is 25%. It fills soil pores not occupied by water. Anywhere there's no water, then there's hair. Provide oxygen for roots, respiration, and microbial processes. Then the living organisms, like I said, is less than 1%, and it includes bacteria, fungi, actinomycetes. Yes, in those days, the actinomycetes used to be called fungi bacteria, but now they are fully classified as bacteria. So you have the bacteria, the fungi, the actinomycetes, the algae, the protozoas, and then the nematodes. Even though we are talking about microorganisms, but we cannot leave out these worms. The nematodes, as they burrow the soils, they help in encouraging the soil texture, space, and all that. So we have nematode, heartworm, crabs, and other large organisms that may be occupying some parts of the soil. So what are the functions of the soil? The soil is a plant growth medium. They provide support, water, and nutrients for plant roots. They are involved with nutrient cycling. That is, the soil facilitates the breakdown and recycling of nutrients that are essential for ecosystem. Of course, you know some of those nutrients that are cycled, like the carbon, potassium, nitrogen, phosphorus, all those cycles. Water filtration and storage is another function of the soil. The soil regulates the flow and storage of water in the environment. Yes, it encourages the flow and storage of, otherwise, if it does not control it, what would have happened? Maybe, I imagine at times, what actually stops that lagoon? Have you ever been to the lagoon front? You are a UNILAG student. I stay by the lagoon front and look at this water is just flowing up and down. I have bent before and look at what is stopping the water from overflowing where it is. So have you imagined why you have an island and the water does not overflow where it is? So the soil perform regulations of the flow and storage of water, even in the environment. They are habitats for other organisms. The soil is a habitat for other organisms. They support different microbial and macroorganisms. They also regulate the climate because the soil acts as a carbon sink and influences greenhouse gas emissions. How is soil formed? How is soil formed? Soil formation. Am I too fast?
No, ma'am.
All right.
No,
Are you
ma'am.
all right? Okay. So soil formation. Soil forms through the process of weathering of parent rock material. You already know that. And the accumulation of organic matter over time. You know, as trees are falling, leaves are falling, grasses are dying. During dry season, they will die, then they will be buried. Rain will come and they will get pushed into the soil over years, over time. These things get accumulated, and then you have soil. So the processes are influenced by parent material. You have the original rock or sediment somewhere. We have rocks, you know, we have sediments that are the original parent material, like where other soils can develop from or can come from. Then you have the climate that can also influence soil formation, like temperature, precipitation, depending on the nature of the temperature and precipitation, it can affect soil formation. Biological activity, contribution of plants, animals, and microbes help in soil formation. The topography of the material, of the parent material, you know, when you go to part of the east, you see some places you have some gallow, you have some valleys. So soil can know those kind of a place can, weathering can occur due to different activities like flooding, like leaching, like excessive temperature, precipitation. They can affect, you know, the topography can affect the slope and drainage patterns of those kind of soil. Time, the length of weathering and organic matter formation or accumulation will determine soil formation. But that is what happened over time. That is how soil is being formed over time. If you are a geographer, you would have understand more about this, but I know we did some for those of you who did agriculture. So back to business, soil microorganisms of agronomical importance. We are not just talking about all kinds of microorganisms in the soil. I'm sure if you take a gram of soil and pull, maybe you take 1 gram from Unilag, 1 gram from maybe Okokomaiko, you pull them together and you analyze, you will see thousands upon thousands, especially if you use the metagenomic, you will see thousands of different microorganisms. But when it comes to their ecological roles, it is not all of them that are playing important role in agriculture. So we are going to look essentially about the organisms that play important role in agronomy. So the soil processes that are mediated by organisms whose activities are greatly influenced by soil physicochemical conditions and by ecological interaction. So majorly we have plant growth promoting fungi, plant growth promoting rhizobacteria, but those are not all. On this table, you have the microbial groups that you can find in soil that play major roles in agronomy or in agronomical activities. Bacteria is the most abundant among them. And I've only showed some samples here and the functions that they performed. For example, we have the rhizobium, the azotobacter, pseudomonas, and the bacillus. What function do they perform in this soil? They are involved with nitrogen fixation, and you know the importance of nitrogen to soil fertility. They are involved with decomposition of organic matter. So if animals die, a cat, a rat, human, whatever dies, and they bury them, what happens? They get decomposed over time by microorganisms. Then some of these organisms, for example, the bacillus, can suppress disease of plants. We have fungi or fungi. Examples: Aspergillus, Penicillium, and Trichoderma. You can find them in the soil, and they are important for agronomy. What function do they perform? Decomposition of complex organic materials, e.g., most of the time, the fungi will degrade cellulose. The lignin aspect of a plant is the most difficult to degrade for bacteria, for some bacteria. So if you have some fungi like the Trichoderma or the white rot fungi, they can degrade the lignin aspect. And when they degrade the lignin aspect, other organisms like bacteria and other, they will penetrate that plant to begin to take up the cellulose and the sugars that are easy for them to take up. Some of these fungi also form relationship or association with plants. We call such fungi and plants association mycorrhizal association. That is the relationship between the fungi and the roots of the plants. That relationship or association is what is called mycorrhiza. And you have different types of mycorrhiza, about 4 of them: vesicular association, you have the conoid and all that. I think we'll still get to that later. Then this mycorrhiza, what they do is the fungi wound themselves around the root of the plant, especially during the droughts or unfavorable season. And since fungi have hyphae, they wound their hyphae around the root. So their hyphae form a kind of extension that goes down into the root to take water and other nutrients and then pass it to the root of the plant for plant take up. It's an association. So the fungi also benefit from the plants by obtaining cellulose or sugars that they use in their metabolism. The actinomycetes, for example, streptomyces is one of them. Actinomycetes, by the way, streptomyces is what I studied in my PhD. Those were my major organisms, and I still have them since 2008. I'm still using them till now. They are very good with decomposition of organic compounds in the soil. They also produce antibiotics. The algae, for example, the chlorella and the nostoc, carry out photosynthesis and oxygen production within the soil. We have the protozoa, e.g. the amoeba and the paramecium. What do? They help in the regulation of microbial populations, because sometimes you have excessive heterotrophic bacteria in the soil. So amoeba and paramecium feed on some of them. This could be an advantage. It could also be a disadvantage in some situations, but they are important, whether it is advantage or disadvantage. That is why they are very important in agronomy, because they help in regulating microbial populations by feeding on bacteria and fungi. Of course, we have the nematode. And why I'm mentioning nematodes, because what comes to our head when we hear nematode is that they are worms. But we do have parasitic plant nematode. For example, the meloidogene. It's a plant parasitic worm. So while some of them are free-living, some of them are parasitic, they also carry out predation on soil microorganisms, and they help with nutrient cycling. So we'll now look at some of these processes, and I think I will just give maybe one example, because we still come across the other ones in the next topic. So soil processes powered by microbes of agronomical importance. I've mentioned this, but I'm going to just list them again for ease of understanding. Decomposition of soil organic matter is one role. Nitrogen fixation, phosphorus solubilization, potassium solubilization, and then the production of siderophores in the soil. These are activities or processes that are of agronomical importance. So let us look at them one after the other. So this is the summary, because I might not go through every one of them. So this is the summary. You have decomposition of organic matter, where microorganisms break down plant and animal residues into simpler organic compounds. Here, after this breakdown, what are the end products? We have the humus, which improves soil structure and nutrient retention. We also have the soil organic matter decompositions that are a key to achieving good crop yields. The nutrient cycle, conversion of nitrogen into usable forms for plants is one of them. We are talking about nutrient cycling, and we are looking at the important ones that are important to agronomy. So we are mentioning the nitrogen cycle, phosphorus cycle, and the sulfur cycle. So what happens with nitrogen cycle? Conversion of nitrogen to usable forms for plants. What are the key processes? You have heard severally about nitrogen fixation, nitrification, denitrification, and ammonification. So if you have forgotten, simply carry your BSc or go online and refresh your memory. The microbes or microorganisms that are involved with these key processes are rhizobium, the nitrosomonas, and the nitrobacter. For the phosphorus cycle, what happens? Phosphorus are available, but they are locked. They are there in the soil, but they are not available to plants. So we need some microorganisms to unlock or to solubilize them so that the plants can make use of them. So solubilization and mineralization of phosphorus is carried out by organisms such as Pseudomonas and Penicillium, the fungi. Then the sulfur cycle. This is where oxidation and reduction of sulfur compounds takes place in the soil. And the microorganisms that is involved is Thiobacillus. Some other processes takes place in the soil that are involved with microorganisms. For example, biodegradation of pollutants. Our farmers, they make use of pesticides, fungicides, and other chemicals. So, we are talking about soil pollution. These pesticides take long time for them to get out of the soil or to be neutralized. So the microorganisms help in degrading the pesticides or the hydrocarbons or other heavy metals that found themselves in the soil or the farm, since we are talking about agronomy. These organisms also help in the formation of soil aggregates. For example, the fungal hyphae and bacterial exudates, that is what they give out during their metabolism, help bind soil activity. For example, when bacteria attacks dead leaves or something, one of the products is maybe organic acid. That is one of the products. So this will help bind soil particles and improve aeration and water retention. What are the conditions? Now we are looking at the composition. What are the conditions that may affect the composition of organic matter? The rate of decomposition influenced by environmental variables include soil temperature, moisture, oxygen, nitrogen content, the quality and quantity of available carbon substrate. So, for example, temperature. If we have dead organic organisms, for example, in Nigeria, which is a region that have high temperature, right? And then you go to temperate region, tropical region, temperate region. Can somebody unmute and tell me, in which region will you have a faster decomposition? If you want to tell me, you can unmute.
High temperature. High
I'm
temperature.
asking the temperate region or the tropical region.
Temperate region, ma'am.
Do you know the meaning of temperate? Goodness Kukoyi, temperate region is where you have cold weather and you are in the tropic, right? Tropical region like Nigeria, like West Africa. You have high temperature, there's heat. So if we bury a dead cat or a dead dog in Nigeria and then in a temperate region, the one in Nigeria will decompose first because of the high temperature. Decomposition is affected by temperature. The higher the temperature, the more or faster the decomposition will take place. Moisture, it depends. If you are talking about, of course, organisms require water for them to carry out their activity. Bacteria loves water, so they thrive more where the moisture is high, but not the same thing for fungi. Fungi just need a little bit of humidity or humid. That is why when you put your belt in your wardrobe, maybe you are out and then rain fell, then you got into your house, clean your shoe, and then store it in your cupboard. In the next 5 to 6 days, you see whitish thing growing on it because just little humidity or little amount of water is okay for fungi to thrive. For bacteria, they require larger amount of water. So the moisture will also determine what will happen. The same thing with oxygen, because some of these bacteria or fungi, some of them love oxygen, some of them don't mind whether there's oxygen or there's no oxygen, they can adjust. So we have the aerobic, the anaerobic, and then the facultative aerobic or anaerobes. So the nitrogen contents will also determine, decomposition, as some of these organisms require nitrogen, as nutrient for them to be able to carry out their metabolic activities. The quality and quantity of available carbon substrate. Carbon substrate is that which they are trying to decompose. So if it is a fallen leaf, for example, that is a carbon substrate. If it is a dead dog or a dead animal, that is the source of carbon. So it will also affect the rate of decomposition. So, but generally, early stages of decomposition are governed by bacterial activity. And these bacterial activities will be affected by some of those factors or variables that we mentioned. Fungi, for example, on the other hand, may prefer more recalcitrant residues. Like I said earlier, if you have plants that have high amount of lignin, most bacteria will not have the capacity to degrade it. Some may, but some may not have the capacity. But when fungi sees recalcitrant, that is difficult to degrade material, some of these fungi likes it, like the white rot fungi. So if you have, that is why you have some fungi that can eat up rubber. They can eat rubber. They can eat whatever you think, nylon, rubber, plastics. So when you have plants, because different plants are available, the amount of lignin is that component of the plant that make it rigid. So lignin differs with different plants, but where you have recalcitrant residues like that, fungi may prefer it. So they may dominate the early stage, or at times when the other bacteria are eating the parts that are easy to degrade. Fungi will take up the difficult or recalcitrant and then decompose it at the later stage. So it is very obvious that a community higher in species of richness is also more diverse with regard to decomposition. What this is trying to say is, if, for example, in a particular community, for example, you have only bacteria, let's assume that in a specific soil environment, it can never happen. You have only bacteria, or a specific species of bacteria is the most abundant. That means once they have decomposed whatever they can take up, they will leave the rest. But if the reason why you have different species, that is species richness. When you talk about species richness, that means you have diverse group of organisms. You have different species of bacteria, you have different species of fungi. So they can come together to work together to decompose. So what this one can take up, this one may not be able to take it up. Another one will take it up because you have different species within that environment. Is it clear, please? Do you understand?
Yes, ma'am.
All right. So most research findings suggest that decomposition activities is not affected by increase or decrease despite species richness. That is, when you have species richness, you have different species. It is not whether the organisms are increased or the organisms are decreased that will affect the decomposition. It is the richness of the species, different species that can do different activity. That is why metagenomics is very important. When you do, maybe whole genome sequencing and all that, and you try to see the different DNAs, the different species of organism that is in that particular environment that you are studying, you will see it, and it will attach the role that they play. Those that can carry out hydrogenation, you will see it. Those that can carry out different processes, you'll be able to determine it. So you'll be able to attach role to the different organisms in different soil environments. Okay, so we are talking about relationship between fungal and bacterial decomposer diversity and organic matter decomposition. In the previous slide, we are talking about species richness, having the essence of having different species in that soil environment. So this table, where some researcher Jiang et al. In 2008 carried out some experiments over they tried to find out what is the relationship between fungal or bacterial decomposer diversity and organic matter decomposition based on experimental studies using single or complex compounds. Single, that is, they determine the carbon source. So for the single carbon source, they use cellulose. And for this complex carbon source, they use several compounds. So compounds that are complex, like maybe wheat straw, oak tree, and barley straw, wood, those ones have different components. For example, wood will have lignin, it will have cellulose, it will have hemicellulose, and some other things. So let's look at one or two of what they have on the table for Jiang et al. 2008. They mentioned some decomposer types. For example, fungi. They did not tell us the name of the fungi. But these fungi, in the very environment tested, they have 1 to 2 species. And the substrates they added for the experiment is a single type of substrate, that is the carbon source, cellulose. The process they measured to see the level of decomposition, they mentioned cellulose mass loss. What that means is, for example, if you read that paper, you will see the detail. But for example, let's assume they fed this fungal species, just 2 species, Maybe they gave them 5 grams of cellulose for day 1. How did they monitor the progress? Maybe after 24 hours, they want to see, have the cellulose reduced? Maybe by the time they measure, the cellulose has reduced to 8 grams. After 48 hours, maybe it has reduced to 6 grams, and they keep measuring like that. What result do they had? They discovered that the relationship was positive. For the 2 different fungal species, they worked together to reduce the cellulose mass. So there was cellulose mass loss. So they were working. They also tried 1 to 5 species. They used the same cellulose. And what did they measure? They didn't measure cellulose loss this time. It's oxygen consumption that they measure. Oxygen consumption also indicates that cellulose is being taken up. So they discovered that it was strongly positive. Then they tried bacteria, 1 to 8 species, using single cellulose, and they measure cellulose mass loss. They also discovered positive. Then they tried 1 to 72 species. They used a complex substrate, the leaf disc, and they measured carbon dioxide production, which is carbon dioxide production is an indication of that leaf disc is being decomposed, because when it is decomposed, the carbon will be released as the end product. So it was positive. But if you look at this fungi here, 1 to 5 species, and the substrate was just soil, and the soil contains different things. They measured oxygen consumption, but it was weak. The degradation was very weak. This is because probably this soil does not have enough carbon source for the organisms to take up. Look at this fungi. They use 1 to 8 and is a alder leaf. They measure leaf mass. Despite the fact that you have 8 species there, it was neutral. There was nothing, no activity was taking place. If you look at the last one, you have 16 species. So it is like the statement was saying in the previous, it is not how plenty the species are. It is not how plenty they are. It is when you have different species that are able to perform. So despite the fact that these ones have 16 species, and they are given a very strong substrate, which is a complex substrate like wood, they measured the wood mass, but they saw that there was no activity. They could not take up this wood mass. So the biochemical processes in soil. Biochemical processes in soil. Soil is a dynamic system where biochemical processes are driven by microorganisms, and then their metabolites or metabolic products are critical for maintaining soil fertility, nutrients availability, and health of the ecosystem. There are times that people plant, you plant agronomical, then the soil becomes depleted. But in agronomy, as much as you want soil, a good product, you want your plants to thrive, they are also monitoring or managing the soil health. The ecosystem must be healthy. So the processes, we have enzymatic activity in soil. Enzymes produced by microorganisms catalyze critical soil reaction. This is vis-a-vis the processes that we have mentioned. We mentioned degradation, right? Decomposition. What do you think are the biochemical processes that help that decomposition? That is what we are talking about. It's because of the enzyme activity of those organisms within the soil. So when decomposition takes place, what happened? The microorganisms were able to produce enzymes that are able to react in that soil. For example, urease, cellulase can degrade the cellulose. If those ones that took up the cellulose, if they don't have the enzyme cellulase, they cannot degrade that cellulose. The urease helps in degrading urea in the soil. Then you have the dehydrogenase enzyme. All these help in decomposition. So these are indicators of soil health and fertility. So when you carry out metagenomics, maybe the proteomics of a soil sample, for example, you are going to see the different enzymes. If you check for the enzymes using metagenomics and you're able to find cellulose, you know that definitely for you to find the cellulase, that means you have organisms that are degrading cellulose in that environment. The kind of enzymes that you see in a soil sample when you analyze them will tell you the kind of activity that is taking place. If I see cellulase, I already know that cellulose is available and organisms that are taking up cellulose are there. If I see proteases, protease enzyme, I know we have protein in that soil sample, and that is why the organism is releasing that enzyme to be able to take it up. And when they do this, they maintain the health and fertility of the soil. So during decomposition, what else happens apart from the enzymatic activity? We have production of soil organic acid. Organisms can release organic acid as their metabolic products. Acids like the citric acid, oxalic acid, that are nutrients for other organisms to use as their own nutrient, or for plants to uptake as plant nutrients. Decomposition activity also leads to siderophore production. Microbial compounds that chelate ion, they enhance plant nutrients acquisition within the soil. We'll see more of that. So the biochemical processes that takes place during the composition of organic matter in the soil is what I have just told you. So the enzymatic activity in soil, role of enzymes in soil, role of enzymes in soil. Soil enzymes are biological catalysts produced by microorganisms. The enzymes facilitate and accelerate chemical reactions in the soil. You already know that from secondary school. And they contribute to nutrient cycling and organic matter decomposition, as I discussed in the previous slides. Enzymes are very essential for soil fertility as they mediate the transformation of complex organic and inorganic substances into forms that are available for plants. So we mentioned some examples that are useful in the soil. The urease function, break down urea into ammonia, and you know how important ammonia is to soil fertility for plant growth. So the urease breaks down urea into ammonia and carbon dioxide. What is the importance of this? This facilitates nitrogen mineralization, making nitrogen available for plant uptake, ammonia, NH3. So the available nitrogen is in form of ammonia that plants can uptake. Cellulase is another useful enzyme for agronomy. Function. Cellulase decomposes cellulose, a major plant cell wall component. Cellulase will decompose cellulose into glucose. And what is the importance of this in agronomy? They aid in organic matter breakdown and humus formation. Humus is very, very necessary for soil fertility. We also have dehydrogenase. What is the function? Dehydrogenase catalyzes oxidation reduction reactions in soil microbial metabolism. The importance, indicator of microbial activity and overall soil health. So, production of soil organic acid. Soil organic acids are the low molecular weight acid produced by microorganisms and plant roots during decomposition of organic matter. And I've mentioned some common examples like the oxalic acid, the malic acid, the acetic acid, the citric acid, the gluconic acid, and all that. What are their functions? What are the functions of soil organic acids? 1, nutrient solubilization. Organic acids bind minerals like calcium, magnesium, phosphorus, and iron, helping to convert them into soluble forms that plants can absorb. Example, oxalic acid and citric acid can dissolve in soluble phosphates, making phosphorus available for plants. What other function do organic acids perform? They help in the regulation of soil pH. The release of organic acids can locally acidify the soil, improving nutrient availability in alkaline soils. Another function they perform is facilitation of microbial interactions. Organic acid attracts beneficial microbes. It's like it's like you put a piece of sugar, you see different hands coming out from different directions to want to partake of the sugar. So when you have organic acid, it attracts different beneficial microbes to the rhizosphere, that is the root zone of the plant, hereby enhancing microbe-plant interaction. Okay, so Olakunle Adetono is asking, Ma, will you give us this material? So I want somebody that was in the class when he was away answer that question. You can unmute and answer. Boluwatife, what did I say about the materials?
Yes, ma, you said material will sent to us after the presentation.
Exactly. All right. Any questions so far? Okay, so now let's look at siderophore production. Siderophore production. What are siderophores? Siderophores are specialized low molecular weight compounds secreted by soil microorganisms, particularly bacteria and fungi, under ion limiting condition. Take note that it is not all bacteria or fungi that can produce siderophores. It is not all fungi and bacteria that can produce siderophores. But those that can produce siderophores will produce them when there's limitation of iron. When there's need for iron and there's no iron in the environment, these siderophore producers will perform their duty. So they bind or chelate iron from insoluble or unavailable forms within the soil and make it accessible to other microbes that need iron and plants. I hope you understand that. So what are the functions of siderophores in the soil? So essentially what I'm saying is, where you have limiting iron around in the soil, but there are iron that are natural, that are locked up somewhere within the soil. We have organisms, bacteria or fungi that can bind iron from their insoluble form within the soil and make it available to microorganisms and plants that are unable to pick it up. So functions of siderophores in soil: ion chelation, that is binding of iron. Siderophores bind ferric iron, that is iron 3+, from soil minerals, converting it into a soluble form, iron 2, in bracket, that plants and microbes can absorb. You know, iron is very important, very essential for processes like photosynthesis, respiration, enzyme functions in plants and microbes. So if they don't have sufficient iron, the processes might be deformed, might not take place appropriately. So for organisms like microbes, some microbes and plants to be able to utilize iron, that ion must be in form of iron 2. They also help in plant growth promotion. Siderophore producing microorganisms enhance plants growth indirectly by improving ion availability and suppressing pathogenic microorganisms in the soil. Suppression of pathogens. Siderophores they outcompete harmful microbes for iron. You know, when you have organisms, different species of microorganisms in an environment in the soil, and there's depletion of iron, those organisms that are not siderophores will be outcompeted, even though they are harmful microbes, but because they are, it's like, lion now. Lions like to swallow anything that is, what do you call it now? But when lions are well fed, they will be looking, they won't be able to eat up any prey. But in a situation where there's scarcity of prey, and you have some lions that are, that have the requirements, they are healthy, they can outcompete a weak lion because it does not have the nutrients and the strength to fight. So also when you have species of microorganisms in an environment, and then iron becomes limiting, there's no iron, and iron is needed for strength and to carry out metabolic activity. The microorganisms that can produce siderophores will definitely outwit the harmful microorganisms, because the harmful microorganisms do not have the capacity to take up the iron in the environment. So example of microorganisms that can produce siderophore include Pseudomonas, Bacillus, Azotobacter, and then the fungi include Trichoderma and Aspergillus. What are the importance of these biochemical processes that we just talked about under this decomposition? Biochemical processes collectively drive the nutrient cycling essential for plant growth. They enhance soil structure and fertility, maintain a balanced and healthy soil microbial ecosystem, support sustainable agriculture practices by minimizing the need for chemical fertilizers. Then, when you try to understand the processes that we have just talked about under the composition, it helps you as an agronomist or agriculturalist in adopting practices such as organic amendments instead of using chemical fertilizer that will destroy the soil. So you can opt for organic amendments, crop rotation, use of biofertilizer to maintain and enhance soil productivity. So what are the impacts of agronomic practices on soil microbiology and biochemistry? What are the impacts? Tillage. What we are saying is, how does these agronomic practices affect soil microorganisms or soil microbiology and the biochemistry? Tillage. You know what tillage means? When farmers till the soil. When you keep using hoe or cutlass or machine, whatever, to till the soil, that activity alters microbial habitats. They displace microorganisms from their natural habitat. So they alter microbial habitats and population dynamics. What about fertilizer applications that farmers do? Excessive use of fertilizer may suppress beneficial microorganisms and enzymatic activity in the soil. Crop rotation and cover cropping enhance microbial diversity and nutrient cycling. Organic amendments, that is manual or compost formation. You know, manual like use of cow parts and all that. Compost is when you put together all these dead plants, and then you leave them for a long time until they become humus. You can read about compost formation. This activity promotes microbial growth and enzymatic activity. So summarily, what is happening here is that how agronomic practices affect soil microbiology and their biochemistry or their enzymatic activity. Tillage and fertilizer application affect negatively microbial habitat, microbial population, suppresses the beneficial microorganisms and their enzymatic activity, while crop rotation and cover cropping or organic amendments like manure and compost promote microbial growth and enzymatic activity. So, what are the environmental factors that may affect soil microbes? We have mentioned this earlier. Soil pH. We have different soil. The pH of some soil are high, some are neutral, some are low. The soil with low pH are acidic soil. The soil that have high pH are alkaline soil. And I think plants require like neutral soil for proper growth. Soil pH influences microbial community because there are some microorganisms that cannot withstand excessive low pH like acidic medium, while some prefer. So that is why we normally carry out experiments to optimize the environmental condition that microorganisms need for their proper activity. So you'll be able to determine, so microorganism prefer low pH, that is, prefers an acidic soil. These organisms prefer alkalinity or alkaline soil. This organism prefer soils with neutral pH. So you have to carry out those experiments. That is why the research institutes are there. Moisture content also determine microbial mobility and nutrient solubilization. Temperature affects metabolic rate of soil microbes. Different microorganisms react to different temperature. We have microorganisms that can withstand high temperature. We have some that prefers low temperature. We have some that are just neutral. Oxygen availability, I mentioned that, is very critical for aerobic organisms and for microbial processes. For decomposition, aerobic condition is highly necessary. Application of soil microbiology and biochemistry in agriculture. We apply soil microorganisms in forms of biofertilizers, use of microbial inoculants to enhance nutrient availability. It is common now. We have several research institutes that put together different rhizobial species and use them as biofertilizer. There's one in IITA in Ibadan, which they use to enhance the growth of legumes, beans, groundnut, and all that. What they do is to grow different species of rhizobia in their lab, and then they use it to coat the beans or legumes, the groundnut, before they grow. And they discovered that this actually enhanced the growth of these legumes by 40%. Biopesticides are also there. Instead of using these pesticides, chemical pesticides that persist in the environment, that causes environmental degradation, there are some microorganisms that can be used to suppress soil-borne diseases. For example, trichoderma. You also have some bacillus, like Bacillus thuringiensis, that can be used to produce pesticides. Then soil remediation. Microorganisms can be used to clean up contaminated soils. I have a paper like that where we deliberately contaminate, we mimic the farm environment in the lab and deliberately contaminate it with crude oil. And when there's crude oil, plants will not grow on that soil. But there are some organisms you can use to inoculate that soil and give it like few weeks or months. After that, if you try to plant your plants there, they will grow because the organisms have already cleaned up the oil there. That is bioremediation. Microorganisms can also be used for sustainable agriculture, like leveraging microbial processes to reduce chemical inputs. And that will also help us to maintain the environment, which is where the world is going now. Chemical processes, because of the nature of the degradation they cause on the environment, people are now going like eco-friendly products. And microorganisms are also very important when it comes to eco-friendly products. So in conclusion, microbiology and biochemistry of soil processes is fundamental to enhancing soil fertility, crop productivity, and environmental sustainability. Farmers and researchers must adopt practices that conserve and promote beneficial soil microbial activities to ensure long-term agronomic benefits. So we just finished the aspect of decomposition. We mentioned the activity of agronomically important microbe, the function in decomposition. We know the function in nutrient cycling, and we mentioned the different nutrient cycling. So we want to look at the nutrient cycling. And I think I will just mention nitrogen fixation. You will read about nitrification, denitrification, and ammonification on your own. Just bring it back to your memory. So nutrient cycling, which is one, nitrogen cycle. I mentioned that we are going to look at 3. Nutrient cycling, we look at nitrogen cycle, we look at phosphorus, and then I think I talked about, I've forgotten the third one, maybe sulfur. Under nitrogen cycle, you have key processes. The major one is nitrogen fixation. We also have nitrification, denitrification, which is the opposite of nitrification, and then ammonification. The microbes that are involved in these processes is Rhizobium, Nitrosomonas, and Nitrobacter. So let's look at nitrogen fixation. Nitrogen fixation is the biochemical, or before I continue, any question? Any question? All right, so nitrogen fixation is the biochemical process through which atmospheric nitrogen gas, N2, is converted into ammonia, NH3, or other biologically useful forms. That is useful for plants. Don't forget that in this course we are talking about soil plants, okay? That will be useful for plants. The processes are carried out by a group of microorganisms called diazotrophs. And I mentioned Rhizobium, Azotobacter, Clostridium, and Cyanobacter or Cyanobacteria. Nitrogen fixation is very, very important because atmospheric nitrogen, that we have up to 78% in the air are not useful for plants because they are not available for the plants. So they have to be fixed. Before they can be useful for plants. And one of the group of organisms that can help are this Rhizobium, the Clostridium, the Azotobacter, and the Cyanobacter. So nitrogen is very important for plant growth. And this form of nitrogen can be fixed abiotically, that is, through some other physical processes, or biotically. But the biotic nitrogen fixation is the most important fixation. And because we are talking about biology, we are going to look at the biotic fixation. So types of nitrogen fixing microorganisms. Types of nitrogen fixing microorganisms. We have symbiotic nitrogen fixation, which occurs in association with host plants. Example, Rhizobium species in legume nodules. You can read about nodule formation. We have the free living nitrogen fixation that are performed by non-symbiotic microbes. Example, Azotobacter and Clostridium. Then we also have associative nitrogen fixation, which occur in close association with plant roots, but not within root nodules. Remember that the symbiotic nitrogen fixation, symbiotic with the root nodules, but associative nitrogen fixation occurs with the plant, but not within the root nodules. Example, Azospirillum. Then the cyanobacteria fix nitrogen in aquatic and terrestrial ecosystem. Example, Anabaena, because they are able to form heterocysts. What are the significance of nitrogen fixation? What are the significance of nitrogen fixation? They make nutrients available to plants. They convert inert atmospheric nitrogen into ammonia. Ammonia is the form that is useful for plants for synthesizing amino acid, protein, or nucleotides in the plants. They are also agricultural important. They are of agricultural importance. They enhance soil fertility naturally, reducing the need for synthetic fertilizers. So if there's good nitrogen fixation, there will be no need for synthetic fertilizer, or there will be a reduced need for synthetic fertilizer. Nitrogen fixation also have environmental impacts. They play a critical role in the global nitrogen cycle, maintaining ecosystem balance. So the biochemical process is a cornerstone of life because nitrogen is a fundamental component of biomolecules. Why? Because that is what helps in the formation or synthesizing of amino acid, which is the precursor of protein and nucleotides for all living organisms, whether plants or animals. So the key steps in biological nitrogen fixation. Nitrogen fixation is catalyzed by nitrogenase enzyme. That enzyme is called nitrogenase. So what happens? The enzyme nitrogenase is central to nitrogen fixation. It catalyzes the conversion of atmospheric nitrogen, N2, into ammonia, NH3, under anaerobic conditions. The reaction requires a large energy input as nitrogen has a triple bond that is highly stable and difficult to break, that is N2. And the reaction, the chemical reaction, is therefore UN2 plus 8NH plus 8 electron and 16 ATP adenosine triphosphate, and then the arrow. On top of that arrow is where you will have the nitrogenase, showing that it is the nitrogenase enzyme that is catalyzing the process. Then the product will be 2 molecules of ammonia plus hydrogen and 16 adenosine diphosphate. The energy required, 16 ATP molecules, are consumed for every molecule of nitrogen that is fixed. Then the ATP is provided by cellular metabolism of the organism, often through processes like photosynthesis or respiration in the host organisms. The electron donor system, the electrons are transferred into nitrogenase via electron donor, such as the ferrodoxin or the flavodoxin. These donors are reduced during cellular metabolic reactions. And this diagram is just showing you nitrogen, what nitrogenase enzyme complex look like. Look at this enzyme in an environment that is anaerobic. The nitrogenase enzyme complex consists of two main components. So look at the components on the left side and the components on the right hand side. So the component 1, dinitrogenase reductase, that is iron protein. This is the iron protein. So you see iron 3 up, iron 2. Iron 3 has to be converted to iron 2 for it to be useful for plant uptake. So these are the donor, ferrodoxin. Ferrodoxins are the donor, you know. So dinitrogenase reductase, that is iron protein, transfers electrons from the donor ferrodoxin or flavodoxin, either of the two, to dinitrogenase enzyme. Then it requires ATP hydrolysis for each electron that is transferred, and we are talking about 16 ATP. Then dinitrogenase molybdenum ion and sulfur cluster is the second aspect of the complex, which is on the right-hand side. You see the ion, molybdenum, and sulfur protein here on the right-hand side. So they catalyze the actual reduction of N2 to ammonia. So they produce ammonia from N2, from nitrogen. They fix it and convert it to ammonia and releasing hydrogen. So this ammonia is what the plant will uptake. Nitrogenase is highly sensitive to oxygen. So that is why it happens. The complex happens in an anaerobic environment. Using protective structures like heterocysts in cyanobacteria. If you look at the last lecture we had, here, types of nitrogen-fixing organisms. So in cyanobacteria, for example, Anabaena, they can form heterocysts. So therefore, under anaerobic condition, they can also fix atmospheric nitrogen, producing oxygen scavenging proteins like hemoglobin in legume root nodules. So you have to read about the different type of nitrogen fixation, but this is what the enzyme nitrogenase complex look like. And nitrogenase can only take place during anaerobic condition. So this is the overall diagram of what nitrogen fixation look like. We know this right from secondary school, but you need to put more flesh to it because this is a higher class. You already know that nitrogen is present in the air. That is why it is called atmospheric nitrogen, but they are not useful there in the atmosphere. They have to be fixed for plants to be able to assimilate it. So you can have it as lightning, for some other methods that are abiotic methods, as lightning and all that. Then when it falls on the plants, you have the nitrogen bacteria in root nodules for, this kind of associative, a kind of association where the root nodules and the bacteria forms relationship to fix the nitrogen. And nitrogen fixing bacteria can fix it directly into the soil by converting it to ammonia like we discussed. So you can see it. Other activity, that was why I said there are some other activity like abiotic, like volcano also helps, but that is not our main concern. We are talking about the biology aspect. So you have ammonia, and then you can see some other processes. When you have nitrification, then you have nitrates, and then nitrification, you already know the names of the organisms, Nitrosomonas and Nitrobacter. Then you have the nitrates that is also useful to plants. Plants can assimilate this nitrate when you follow the arrow, assimilation by plants. So when plants dies, or this animal dies, or their products falls to the soil, you can see the dead animals and dead plants. What happened? You have decomposers like bacteria and fungi that carried out this process, ammonification. Ammonification will convert these dead stuffs to ammonia, which is also taken up by plants through nitrification, and then nitrate is produced in the soil and taken back to the soil. So, but you can also have denitrification. The nitrification will, reduce nitrates to nitrites and then back to atmospheric nitrogen, and the cycle continue that way. So symbiotic nitrogen fixation, this is the diagram, so you can study it on your own. You can read the full paper. Just remind me when I send this slide to you. Let me also send the full paper to you so you can read it, how symbiotic. So you have different type of nitrogen fixation, so you can read about them. So I'm giving you the full paper to read so that you can understand. So the phosphorus solubilization, I didn't want to take you this. I want you to read this yourself. But let me just brush it up. And you look for the circles yourself. Phosphorus solubilization. Phosphorus is an important macronutrient in plants. Soil processes, soil actually have organic and inorganic compounds of phosphorus. Most of them remain inactive. And when they remain inactive in the soil, they are not available to the plant. So the plant cannot take it up. So it has to be made available. And the kind of organisms that help to solubilize phosphorus are phosphorus solubilizer, phosphate solubilizing microorganisms, the PSMs. And they are a group of beneficial microorganisms that are able to hydrolyze organic and inorganic insoluble phosphorus compound to soluble phosphorus that can easily be assimilated by plants. And the example of phosphorus solubilizing microbes are Pseudomonas, Bacillus, Rhizobium, Micrococcus, Flavobacterium, Achromobacter, Alternaria, Aspergillus, Cephalosporium, Cladosporiums, and all that. So you have plants, fungi and bacteria. If you take up any sample of soil, these are the organisms that you are going to isolate. What is the mechanism of phosphate solubilization? So the PSMs apply various approaches to make phosphorus accessible for plants to absorb. How does phosphate solubilization microbes work? One, they can lower the soil pH. Organic acid and proton are released by Pseudomonas to produce gluconic acid. So this will help to solubilize the phosphorus in the soil. They can also carry out chelation. The hydroxyl and carboxyl groups of the acid chelate cations bond to phosphate and convert it to soluble form of phosphorus. Nitric acid reacts with calcium phosphate and convert it to soluble forms. Another method or mechanism is through mineralization. PSMs mineralize soil organic phosphorus or phosphate by the phosphatases, which is an enzyme that can break down phosphates, thereby releasing inorganic phosphate that will be immobilized by plants. And this is the circle. I didn't even know that I've prepared it for you. I didn't have intention of giving you all this information. So you can study this microbial phosphate solubilization flow charts and see how it works and see all the methods that I have mentioned. So you have phosphorus in the soil but are unavailable. So microbes can help you, you can have the inorganic form, you can have the organic form, then the phosphate solubilizing microorganisms will make them available to plants by converting inorganic forms, solubilizing them into organic acid, inorganic acid, H2S production, exopolysaccharide production, and siderophos. We make the phosphorus available. Then the organic forms like phytene, phytase, sugar phosphate, nucleotide, and phosphoproteins. Enzyme activity, enzymatic degradation can occur, converting them to acid phosphatase, alkaline phosphatase, phytases, and all that, and then making the phosphorus available. Then potassium solubilization. Potassium solubilization. I guess you will read this yourself. Oh, Let me see. Potassium solubilization. This is the schematic illustration, so you can read it yourself. Schematic illustration of potassium solubilization by potassium solubilizing microorganisms. So you have the plant growth improvement when exudates help in unlocking the potassium that are locked in the soil and making it available to the plants. Production of siderophores. We have discussed all this. Siderophores are low molecular weight, blah, blah, blah. So siderophore producing microorganisms produce numerous ion chelating compounds, which can cut short plant stress under ion stress condition. Effects exerted by siderophores producing rhizobacteria on plants. This table is just trying to show you the effects of siderophore on plants. That is how siderophore has helped to improve or enhance the performance of plants. All right, any question? It's been a long lecture, but what I just tried to do is to give you an example of how you are going to do your presentation. That's the reason I'm giving you 1 or 2 lectures. Previous years, I gave them their topic straight and it started. So you are supposed to have your topic, you're supposed to have your title, your matric number, and your name. You are supposed to have an outline. All these are info. I'm not supposed to tell you, I'm supposed to just judge you. Introduction, have enough figures to explain. When you have figures and charts, and diagrams, they help you, to explain. And you have to do better than myself, Your slides must not be wordy, In normal slides, you have maximum of 36 words. So, but when you have up to 50 words, it's still acceptable because it will not look too wordy. But when you now put everything and it's too wordy, you are going to lose marks. So try to use tables and figure. All right, so any question before we round it off? Feel free to unmute. Am I talking to myself?
No, ma'am.
No, ma.
No question.
So you guys so quiet like this. Master students, if you come to read abroad, nobody is going to spoon feed you like this. So you'll be giving your topic straight, right, and come and present. So any question, please? So let me ask my question then. So Raphael, what do you think about today's lecture?
Okay, it was insightful and it was well detailed from the slides you've been showing us. And
All right.
it was long as well.
Okay, so Ogunleke, what do you think about today's lecture? When I also asked you about today's lecture, yes, in form of how you feel about the teaching, but 1, 2, about what knowledge did you gain? When I attend any lecture, any webinar, any program, I try to look at the gap that I have filled. I have a knowledge gap. After that lecture or webinar or seminar, did I gain anything? Even when students are presenting, that is what lecturer looks out for. We've come to learn because we believe that by putting that topic together, you have read more than every one of us. Maybe I've read only one or two papers, but you must have read up to 10 because you are compiling that topic. So when I asked about today's lecture, yes, about the lecture then, any knowledge gain about the topic, what do you think, especially in terms of farmers, agronomy, environment, what do you think generally? So, Raphael, Adebola have spoken, right?
No, ma.
Okay, so what do you think? Abi Debola, you have been cooking in the kitchen.
No, ma.
You just
To the role of microorganism in in agriculture, microorganisms help in microorganisms helps a lot in agriculture. For example, to
Hmm.
it helps in sustainability, like the use of biopesticides instead of um synthetic pesticides.
Okay, at least you made. Who spoke first? Was it Raphael?
Yes, ma'am.
Okay, so now Daniel. Daniel, you can unmute.
So the lecture actually went well. We were able to know the functions of soil, the soil formation, and also some microorganisms and their importance. Things like functions of soil, like their plant growth, the nutrient cycling, and the climate regulation. Then I also learned the conditions affecting the decomposition of organic matter.
Hmm. Okay. Raphael.
Yes, ma'am. Here, ma'am.
What's your take?
Okay. During the course of the class, I learned the role of microorganisms in releasing nutrients, some essential nutrients in the soil, because most of the nutrients that are in the soil are in the states they are in. They are not accessible by plants, but when microorganisms come in place, they help to convert them to the soluble forms that plants can access and all that. Yeah, it was all about the role of microorganisms in the soil, general, in agriculture and everything.
Okay. Kukoyi.
Ma'am, thank you for the lecture, ma'am. The first thing I like, apart the major thing I first grabbed was the siderophore production,
Okay.
and also the iron. Okay, the organisms that helps with iron binding, making iron from iron 3 to iron 2. Then also the soil remediation, the example you gave brought another insight. I have known about cleaning up contaminated soil. But when you mentioned crude oil and when we use specific microorganisms that within weeks you should be able to clean up the soil. Before I usually think it will take about 2 months, 3 months, or more than
Yeah,
that.
it depends on the microorganisms. It depends on several other factors that we have mentioned. And it also
Okay.
depends on the capacity of the organisms to produce the right enzyme, and is the environment conducive for that microorganisms.
Good.
It depends if you are using autochthonous microorganisms, that is microorganisms that are naturally within that soil themselves, or are you introducing another microorganism from outside like we did? And also depends on, are there nutrients that are the nutrients sustainable? For example, we just talk about some microorganisms might need iron and there may be no iron. When you read things like bioaugmentation, biostimulation, bioaugmentation, that is outside the original or natural or autochthonous microorganisms that are in that that environment that is contaminated. If you wait for the natural organisms, which are the autochthonous organisms, it may take months or years before they clean up. But when you augment, you bring in organisms that you already know that can take up the contaminant, and you introduce that organism, it will make the process faster. When you carry out biostimulation, that means you analyze that soil environment and see if there's enough nutrient for the organism to carry out the cleanup. Otherwise, you can stimulate the organism by adding the right nutrient that they need to add. So everything you do is more than just talking this way. Everything you do, like we did, if you read down my paper, you will see that we carried out biostimulation, we carried out everything like that, nutrient additions. Okay, so Osofisan, Olakunle, can you just summarize everything we learned today? I gave you a table of summary today.
Hey, good evening, man. Thanks for the lecture. Okay, so my take on this our topic for today is that the component of soil, which are mineral particles that has 45%, organic matter with 5%, water 25%, air 25%, and a living organism with less than 1%. Also, the function of soil, which acts as plant growth medium, then nutrient cycling, water filtration and storage, where we talk about soil having the ability to retain water, not allowing it to overflow. Then it also acts as habitat for organisms. Then climate regulation. Again, the soil formation, where we talked about the parent material being the principal player in soil formation. Then climate, biological activity, where we talk about organisms and the enzymes they secrete, just to ensure the soil itself is good enough for plant growth. Topography, and we talk about timing of the soil as well. Again, then we also talk about the availability of how do I put this now? Nutrient availability, which microorganisms play a large role in fixing it, such like the nitrogen fixation, which is carried out by Nitrobacter, Rhizobium, and the likes.
All
so
right,
I'm actually on transit.
I know, that's why your internet is not stable. Osofisan, do you have anything to add? Boluwatife?
Yes, ma. Good evening,
Good
everybody.
evening there.
Yes,
Yeah,
ma'am.
thank you.
So thank you so much for the class. So I think almost everybody has said quite a lot, and just to touch base real quick, talked about the soil processes powered by microbes of agronomical importance. Talked about the decomposition, nitrogen fixation.
Because I've opened it for you, that this is the summary of
I was not looking at my screen right now, ma.
Okay, so summary of what we did today. Soil processes powered or carried out by microorganisms of agronomical importance. The soil processes are decomposition of organic soil, organic matter, nitrogen fixation, sulfurous solubilization, potassium solubilization, and production of siderophores. So
Yes.
we're talking about the organism being useful for the composition of organic matter, carrying out nutrient cycling, among which one of them is nitrogen fixation.
Yes,
all right. So you people should read. I will send this slide immediately before I forget. So discuss among yourself. Tell me 2 days that you can be available for your ballot. Because if we do this ballot online, there will be bias, because I do it for undergraduate. I will just wake up one night in the middle of the night and put the topic. And usually the first person to see it will take the topic they like. Then they will put it on the platform. I have picked topic 1. My name is so. This. Do you want it that way?
I think we should ballot, ma.
If I put it that way, so the first person to see will be the first person to choose their topic. Then everybody will picking. The last person to see it will pick whatever topic that is left. So you prefer balloting, right?
Yes, ma'am.
Let us ballot,
Yes,
ma'am.
ma'am.
Okay, I will try to speak to one of my PhD students, whoever will be around. So maybe I should first of all get in touch with them, find out when they will be around. But I want you to give me a day or two that you'll be available, so that maybe the day you are coming to school, so you won't have to come to school because of balloting. If you know when you are coming to school, you can let me know 1 or 2 days, but I won't force the PhD student to come to school because of you. So any of the 2 days that the PhD student will be on ground, they will tell me that you just go there. Within 10 minutes, you are done. I've told you 2 things you are balloting for. You pick whatever topic you pick is your topic. Then you also ballot 1 to 6 or 1 to 7. Number 1 is the first person to present. No argument. But I'm going to give all of you 2 weeks to do your work. And in those 2 weeks, there will be no lectures. So next week, Thursday, I will take you another topic. But please let us ballot between now. Today is Thursday. Between now and Monday, let us do the balloting. Or between now and Tuesday, let us ballot. Thank you. Any question? All right. So thank you so much. But you took time to join, despite the fact that I asked for 10 minutes grace to be able to settle down because of the time differences between where I am and Nigeria. I still joined and you people were not here. So please try to adjust. So have a lovely week.
Thank you very much, ma'am.
Thank you very much, ma'am.
You're welcome. Bye.